right, so glycolysis and the citric acid cycle. The first two steps of the process of cell respiration. Um, but if you look at the top of your guided notes, we've got an extra kind of intermediate step in there. We're going to talk about glycolysis, the oxidation of pyruvate, and the citric acid cycle. So we're going to try our hardest to kind of break this down into kind of manageable pieces. Um, also, where to go? There's a lot of intermediate products, as you saw in the book yesterday. Like going through that citric acid cycle, there's a lot of like intermediates. You do not have to memorize any of those. The goal is to know the electron carriers, the outputs, so like ATP, carbon dioxide. You do not have to memorize the intermediates. I didn't take a class where I had to memorize the intermediates. So don't worry about like knowing the actual like bits and pieces of the citric acid cycle. Glycolysis, on the other hand, is a relatively simple process, at least compared to the citric acid cycle. So when we break down glycolysis into like its base words, glyco means sugar and lysis means splitting. Lysis also comes up with like the breaking or lysing of cell membranes. So lysis means splitting. So glycolysis as a whole means sugar splitting. We are literally breaking up sugar into two major pieces. This process takes place just wherever in the cytosol. Just wherever in the cytosol. Usually somewhere close to the mitochondria, but it can happen wherever the enzymes needed are. So while most of cell respiration takes place in the mitochondria, this process just takes place in the cytosol cytoplasm. The big idea here, if we were to summarize glycolysis in one sentence, it would be this bit right here. Glycolysis is harvesting chemical energy by oxidizing glucose and yielding pyruvate. The process overall is 10 steps, but the big word that I want you to like underline, highlight, etc. is anaerobic. And I don't have my fancy annotation thing signed in because, of course, why would I? Thank you. There we are. The big idea here is, or not this, I need this, is this word anaerobic. Goodness gracious. Every time. What does anaerobic mean? Yeah. Without oxygen. Glycolysis does not require oxygen to take place. We don't need oxygen until we get to the electron transport chain, which we'll talk about tomorrow. So, glycolysis is actually the first step in a lot of processes that aren't cell respiration. When we talk about fermentation, glycolysis is often tagged in that as well. Almost all cells that break down sugars use glycolysis, whether or not they do cell respiration in the way that cells with mitochondria do. There are a lot of prokaryotes without mitochondria that harness the energy in glycolysis. So, anaerobic process. What happens is two big phases. We can break down all 10 of those steps into two big phases, and I'll write this out with you. So, we've got energy harvest and energy investment. And we're going to switch those. So, in glycolysis, as with anything, it takes money to make money, right? We need to put a little bit of energy in order to get a lot of energy out. So we call glycolysis overall like a net gain of energy, but we still have to invest a little bit. So in your phase 1, we're going to draw this along with us. Should I? Would you rather me draw over this diagram or create a new blank slide and go from scratch? Scratch? Okay, cool. So I'll flip back and forth, but we'll do that. Beautiful. All right, so we've got energy investment and energy harvest or energy payoff. So we're going to start with phase 1. We're going to start with phase 1. That is energy investment. So this should be the first big box in your guided notes. We're going to start with glucose. Glucose is a 6-carbon sugar. So we are going to keep track of carbons because that helps us keep track of where we lose carbon dioxide later on. But we don't need to know the names of these intermediates apart from like the beginning and the end product. In fact, we're not even going to write out the names of the intermediates. All we need to know is we start out with glucose. We start out with glucose. From there, we are going to invest 2 ATP in order to add 2 phosphates to our glucose. So we are going to invest 2 ATP. So we're going to put 2 ATP in, and out we're going to get 2 ADP, or adenosine diphosphate. And what that's going to look like is our glucose molecule, but with 2 phosphates. That is called like 1,6-fructose bisphosphate. We don't need to know the name of it. We just need to know that we are investing 2 ATP. I always like to put little squiggles or highlight ATP. That way I know where the energy is. Because all of this is all about energy. So, after we've got our intermediate, is a 6-carbon intermediate, going backwards, what happens is that is broken up into 2 3-carbon molecules with phosphates. So that is going to break off into more intermediates. That's phase 1. It took some energy to get started. Doing okay so far? All right. Phase 2 is the payoff. Phase 2 is energy harvest. What we're going to do, going backwards again. We are going to add more inorganic phosphates, not necessarily from ATP, to get yet another intermediate. So from here, we are going to take an inorganic phosphate, and we're going to get something like this. And if it helps, I probably should have said this a little bit sooner. If it helps in these little circles to put little C's, so you know that it's carbon, that's fine. If you want to make a little key that is like a little circle for carbon, I didn't want to put all the C's in there because it'll get kind of messy, at least with my handwriting. But this is our next step. But phase 2 is for energy harvest, and we have not harvested any energy yet. So what happens is we are going to get some electrons off of this molecule. And we're going to do that, and we're going to transfer them to NAD+, and we're going to get NADH. And we're also going to get those phosphates off of these molecules, and we're going to get 2 ATP from 2 ADP. So what's going to happen, we're going to get NAD plus is going to go in, and we're going to get NADH out of that. Make sure you got a little arrow so we know what we're getting. And we're going to take 2 ADP, and out of that we're going to get 2 ATP. And again, that's our we've got energy in ATP, and we've got energy in NADH. Yes. Yes. So we're taking some hydrogens off of this. And that's going to happen to both of these molecules. So same thing over here. We've got NAD plus going in, and NADH coming out. We've got 2 ADP going in, and 2 ATP coming out. Double, triple check. Yes. And in the end, we get, I have to sit on the floor, 2, 3 carbon molecules. And these are pyruvates. So we're going to get 2 molecules of pyruvate out of 1 molecule of glucose. Sorry, it's 3 circles. Sorry. This is the entirety of glycolysis. Now we can always get it a little bit more complicated because it is a 10-step process. We've kind of simplified it a little bit. But this is what happens. We're breaking down a 6-carbon molecule into 2 3-carbon molecules. But we can take all of this process and we can summarize it with this right here. So this is our net process of glycolysis. So this will go in that last little box. We're going to start with glucose. We're going to get 2 ATP out of it. and we're going to get 2 NADH out of it. This simplifies it because if you remember from our last slide, right, we took 2 ATP in, we got 2 ATP out. Those cancel each other out. So we don't write it on here. This is our net process. So if in the little corner of that box you leave a little bit of space, our end products, what we get out of this is 2 ATP, 2 NADH, and 2 pyruvate. This is what we have at the end of glycolysis. So that's glycolysis. Normally, or in a previous bio course or something that's not going as in depth, we would go straight from glycolysis into the citric acid cycle. But there's a little intermediate step because we can't feed pyruvate directly into the cycle. There has to be a little change up. We have to oxidize pyruvate. So, pyruvate is then actively transported into the mitochondria. So we've got our pyruvate from glycolysis. That we can get into the mitochondria. Then everything else will be in that little organelle. So we're going to actively transport mitochondria, or actively transport pyruvate into the mitochondria. It is active transport because we are moving it against its concentration gradient. We want as much pyruvate in the mitochondria as possible. But before we can get to the citric acid cycle, we need to transform pyruvate into something called acetyl-CoA. Acetyl-CoA. That is going to be fed into the citric acid cycle. So if you want to underline acetyl-CoA, that way we know where that word's coming from. And we can summarize this equation. Whoop. Well, I've got some steps, but again, we don't need to know the nitty gritty. What happens is in that pyruvate, we're going to oxidize a certain group, release some carbon dioxide, and then we're going to oxidize those 2 carbons that are left after we release that carbon dioxide, transfer some electrons to NADH, and tag on something called coenzyme A or CoA. So this is what you'll put in your little box. We are going to take our pyruvate. We're going to add on our CoA. This is coenzyme A. We're going to release a carbon. Always, always, always keep track of your carbons to tell you if something's gone wrong. We're also going to get another NADH. Keeping track of these NADHs is really important for our citric acid cycle. Or not citric acid cycle, sorry, electron transport chain. So these NADHs will come back. They're not just byproducts. They're really important. And then we get this 2-carbon molecule called acetyl-CoA. And this is fed to the citric acid cycle while our NADHs will jump down to the electron transport chain. Doing okay? All right. So, started with glucose, got to pyruvate. Now we have moved from pyruvate to acetyl-CoA. We have lost a carbon. So we're starting the citric acid cycle with a 2-carbon molecule. So, here's the citric acid cycle overview. In our citric acid cycle, carbon dioxide is going to be released from our organic intermediates. This is the carbon dioxide that we breathe out. It is the carbon dioxide that comes from these sugars. So the first blank is CO2. We've got some ATP that is going to be synthesized from some ADP and inorganic phosphates. And we have our electrons being transferred to NADH and FADH2. This is our overview. All of the citric acid cycle, we're going to go through the steps and we're going to draw it out. All of those steps are summarized in this little bit right here. We're going to release some carbon dioxide. We're going to synthesize some ATP. We're going to transfer some electrons. That's the goal. It's essentially a furnace that just cranks out these electron carriers, oxidizing and oxidizing and oxidizing these organic fuels. That's why we talked about redox reactions for an entire day, because what we are doing is oxidizing these organic fuels to get the electrons out of them. So, how does this work? First things first, we only get 1 ATP out of the citric acid cycle. For every pyruvate or for every acetyl-CoA that goes through the citric acid cycle, we only get 1 ATP. And we get this ATP from something called substrate level phosphorylation, meaning phosphates are transferred directly to the ATP from our substrate in our enzyme. Most of the ATP that is made in cell respiration is not synthesized through substrate level phosphorylation, which is why we make this distinction. And it's a really long phrase. It just means not using ATP synthase, essentially. So, what happens is our energy is transferred to NAD plus and NADH to get, or FADH, to get NADH and FADH2. Now, this sounds like alphabet soup, I know, but these are our non-energetic electron carriers. These are where our energy is. So, most of our chemical energy is transferred to NAD plus and FADH. That's our second to last dot. And then NADH and FADH2 are our last 2 boxes down here. If you are keeping track of energy, this is where our energy is. Doing okay so far? All right. So, we're going to get into the nittiest of the grittiest with our citric acid cycle. And basically, I'm just going to tell you the steps. So, there's a reason I'm not having you write out each step one by one. And there's a reason why we're mostly keeping track of the electron carriers. So, there are 8 steps. 8 steps to the citric acid cycle. We're going to start with our 2 kind of start points. That is acetyl-CoA and something called oxaloacetate, which I just like saying. Oxaloacetate. If you want to commit any of these intermediates to memory, you do not have to. Oxaloacetate is like the big one. So, because it's in our first step. So, we're going to add acetyl-CoA to oxaloacetate, and we're going to get citrate. Citrate is then transformed into isocitrate by the addition and removal of water. Isocitrate is then oxidized. We can oxidize isocitrate. And we are going to reduce NAD plus to NADH. And after that, I want you to hand write in that we lose a CO2 here. So that's one of the carbons that we've lost so far. We're then going to lose another carbon. And when we do this, we're going to further reduce another NAD plus to NADH. So in the first half of the citric acid cycle, we have already made two, bless you, electron carriers, or energized two electron carriers, and we've lost two carbons. We care about carbons and we care about energy, and that's about it for now. Doing okay? All right. Step 5, our CoA that we added whenever we oxidized pyruvate is going to get booted off for a phosphate group. That phosphate group is transferred to something called GDP, which is guanine diphosphate. It's ATP. It's very, 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 very similar to ATP, only it's guanine instead of adenine. This forms GTP, very similar to ATP, so similar to ATP that we just consider it an ATP. Or we just use it to make ATP down the line. We are left with something called succinate. From succinate, we are going to transfer some hydrogens from FAD to FADH2. So we're going to reduce FAD and oxidize succinate. When we do that, we get fumarate. We're going to chuck a water in there. Get another intermediate we call malate. Then we're going to oxidize malate. We're going to get another NADH. And we're going to regenerate oxaloacetate. We've seen oxaloacetate before because it was the thing that was fed into the cycle in the first place. That's why it is a cycle. Because we add oxaloacetate in and we get oxaloacetate out. And it just keeps going. We're just going to use those carbons to carry out all of these steps in order to get our energy, our ATP, our FADH2, and our NADH. So, I didn't want to make you draw out the citric acid cycle this time. We will eventually. So I've got a little thing for you. This is meant to help you keep track of the carbons. So the number of the carbons is more important than writing the names down. Of course, you can write the names down. So we're going to start with our acetyl-CoA up here. That's what's going to get fed in. Eventually, we're going to get from this, so we've got our 2 carbons in our acetyl-CoA and our 4 carbons in oxaloacetate. And those are going to come together and we're going to get a 6 carbon citrate. Does everybody see where those carbons are coming from? 4 carbon oxaloacetate meets 2 carbon acetyl-CoA. We've got a 6 carbon intermediate. So we've got 4 carbons, 2 carbons, 6 carbons. Further down the cycle, we've got some NADH. That's our first NADH. And we're going to lose a carbon. We lost a carbon. Now we have a 5 carbon intermediate. In the next step, we're going to generate even more NADH, and we're going to lose another carbon. So we're down to a 4 carbon intermediate. We're going to continue moving stuff around and continue harvesting energy. We've got a GTP, which is essentially an ATP. We've got our FADH2, another NADH, and then we get our 4 carbon oxaloacetate back. That 4 carbon oxaloacetate will then get added to this acetyl-CoA molecule, or another acetyl-CoA molecule, not the same one. We've released the carbons here. And it'll go and go and go and go and go, churning out energy as we go. So our net gain here, our net products, what we're getting, 1, 2, 3 NADHs, 2 carbon dioxides, 1 FADH2, and 1 ATP. Now, this is per acetyl-CoA molecule, right? How many acetyl-CoA molecules do we get out of 1 glucose? 2. That means if we were tracking our electron carriers, carbons, and ATPs for glucose, we would have to double these, because this is just 1 churn, and we get 2 turns per glucose, because 1 glucose is 6 carbons. We've released a carbon already, so we get 2 acetyl-CoA molecules. All right. We still got a little bit of time. So, questions here. All right. I want to put it all together. So, you can use a blank sheet of paper. You can use the back of this little thing. I want to help y'all keep track of where this is. And we're going to do this in a very condensed form. So, starting from the top, where do we start? glucose. So, we start with glucose. Our net glycolysis remember was 2 ATP, 2 NADH. We're going to keep track of our energy sources this time. Remember? So we get 2 3-carbon pyruvates. We're going to get 2 ATP and 2 NADHs. Now this is a very simplified version. Remember, because there's that energy harvest phase and that energy collection phase. We're just looking at our net processes. This happens in the cytosol. So we're going to keep track of that by drawing a little mitochondria. Oh. And because it's actively transported, we're going to get a little transport protein in there. Those pyruvates are going to be actively transported into the mitochondria. From there, we have the oxidation of pyruvate. That's our little intermediate step. So we start with our pyruvate. We are going to have NAD plus go in, and we're going to get NADH out. Because we're going to do it for both of them, we're going to get 2 of these. We're going to lose 2 carbon dioxides. And we're going to have to add 2 CoAs. This gives us a now 2-carbon acetyl-CoA. So our net oxidation of pyruvate is going to give us 2 carbon dioxides, 2 NADHs, and we'll have our 2 acetyl-CoAs. Or just tracking our energy carriers. I know that kind of got messy. Y'all tracking this? Do I need to do something different? Okay. I don't have the space up here to go into the citric acid cycle, so I'm going to move to the next slide and pick up there. But you guys can keep going on yours. Close enough. Okay. So we've got our CoAs. Those are our 2 carbon CoAs. They're going to go into our citric acid cycle. It is still in the mitochondria. Very important distinction. Thank you. So, from our acetyl-CoA, we're going to get our 4-carbon oxaloacetate. Whoop. We've got our 6-carbon intermediate. Again, we don't need to know those names of those intermediates. Oxaloacetate's important because it's the revamped one. We're going to lose, we're going to take an NAD plus, and we're going to get an NADH, and we're going to lose a carbon dioxide. So now we have 5 carbons, so 2 carbons, 4 carbons, 6 carbons, 5 carbons. We're going to take another NAD plus, get another NADH, and lose another CO2. And now we have a 4-carbon intermediate. That 4-carbon intermediate is going to have a lot of stuff done to it. So we're going to go from a GDP, or guanine diphosphate, to a GTP. We're going to have an FAD get turned into an FADH2. And we're going to have another NAD plus get turned into an NADH. NADH, CO2, NADH, CO2, GTP, which you could also write out as ATP, FADH2, NADH. But here's the thing, we only did this for one acetyl-CoA, so we are going to have to do this twice if we're tracking it for one glucose molecule. So net from our citric acid cycle, we've got 1, 2, 3 NADH, right? But that's going to happen twice. So it's 6 NADH, 1, 2 carbon dioxide, so it's actually 4 carbon dioxides. One in GTP or ATP, so we're going to get 2 ATP. and 1 FADH2, so we're actually going to get 2 FADH2s. All right. So, add it up all together. Ready? So, our total per glucose, we've got 2 NADHs from glycolysis. We've got 2 NADHs from our oxidation of pyruvate, and we've got 6 NADHs from our citric acid cycle. So we've got 10 NADHs. The only FADH2s come from the citric acid cycle, so we still only have 2 FADH2s. Four ATP and 6 CO2. That's what we get from all of this stuff. These electron carriers we're not done with, though. Those electron carriers will go to the electron transport chain, which we will talk about tomorrow. So, do we remember the net equation for cell respiration? We start with glucose. Remember that? From like the first day we talked about this? With that redox reaction. Six oxygens is going to give us we know where this goes, right? We know where it goes in. We now know where these carbon dioxides come from. Our waters, we'll talk about a little bit tomorrow, but we have used some water in the steps of the citric acid cycle. But this oxygen, that's what we'll talk about tomorrow. So from just this equation, we know where this came from and where this came, or where that goes and where this comes from. Questions? All right. We'll talk about the electron transport chain tomorrow. There is technically a exit ticket on Canvas, but I'm going to push it to bell work tomorrow just because drawing stuff out took longer than I anticipated. Questions here? All right.